Hi, my name is Joseph Ricio, and I'll be discussing RNA splicing. For RNA splicing to occur, we will first need pre-mRNA. Pre-mRNA is obtained through DNA and transcription. In transcription, on the DNA, there is 2 points, a starting point and a Tata box. The starting point is where transcription occurs, and the Tata box is where the transcription factors bind to. These transcription factors are there so the RNA polymerase can bind to the transcription factors and the Tata box, since it cannot bind on the Tata box on its own. Once the RNA polymerase is there, it will then move along the DNA strand so it can make RNA. Once pre-mRNA is made, there's four parts to it. There's an exons, introns, a 5' cap, and a poly A tail. These 5' cap and poly A tails are used so that hydrolytic enzymes do not degrade the pre-mRNA. Okay. So here's a pre-mRNA with the exons and introns. Once it's in this process, SNRPs, which is made of snRNA and protein, along with other proteins, come and make a spliceosome, bending the pre-mRNA. Once the pre-mRNA is bent like this, it will then cut off the introns, and the SNRPs will bind to the nuclear base pairs on each exon and splice them together. Once they're spliced together, the end products are the 2 exons along with the 5' and 3' poly A tail, and leftover intron, which is going to be thrown, and the spliceosome components, which are recycled again into this RNA splicing. Thank you. Hi, my name is Joseph Ricio, and I'll just discuss Operon to you. So, E. coli synthesizes tryptophan and a precursor molecule through a series of steps. Okay, here we have the entire Operon. These are the genes of the Operon and the promoter. Once the RNA polymerase binds to the promoter, it will go down into the trip E, trip D, trip C, trip B, and trip A, and transcribe the DNA of the E. coli and create mRNA, which then will create tryptophan. Now, TRPR is a regulatory gene that creates mRNA, which then codes for a protein, which is a repressor, but this is in its inactive form. Now, when there's a sufficient amount of tryptophan in the system, this inactive repressor will bind with tryptophan to form a structure like this, and this will bind to the operator, stopping RNA polymerase from ever entering on the DNA strand. Hi, my name is Joseph Ricio, and I teach you the lac operon. The lac operon is an inducible operon, which means it's normally turned off. Here on the DNA strand of E. coli, lac I, which is a regulatory gene, creates pre-mRNA. This creates active repressor. This active repressor then bonds to the operator. Because of this, RNA polymerase is not allowed to transcribe the genes of the operator. Now, when a person or animal drinks milk, allolactose, an inducer, causes the active repressor to become inactive, allowing RNA polymerase to bond to the operator and continue and transcribe lac Z, lac Y, lac A. Now, lac Z creates B-galactose sialase, which metabolizes lactose. Lac Y creates permease, which carries the lactose into the cell, and the product of lac A is still unknown. In continuation with the lac operon, RNA polymerase is not able to bind to the DNA strand on its own. It will need the help of CRP. CRP is CAMP receptor protein. When there are low levels of glucose, CMP will bind to CRP in its inactive form and make it active, taking RNA polymerase into the DNA strand and transcribing the genes of the operon. Now, when there is a sufficient amount of glucose, CMP will be in low concentrations. So then CRP will remain inactive and RNA polymerase will have no way of going into the DNA strand.